Thank you for the introduction. It's really an honor to be here to receive the award, particularly with Gero and Peter, who their enormous contributions. It's been a real pleasure to watch here, particularly together. And I want to thank the committee as well, of course, and thank you all for being here. And in the spirit of historical considerations, we can go back even farther, hundreds of years, and the challenges that are faced by neuroscience are similar to those that are faced by psychiatry. In 1809, some of the very first medical language type descriptions of psychiatric disease, what we would now call psychiatric disease, appeared from John Haslam in England, and he described madness and melancholy in words which are essentially identical to how we still describe these today, and the depth of knowledge is comparable as well. For example, what we would today call anhedonia, he wrote, they therefore neglect those objects and pursuits which formerly proved sources of delight and instruction. And this is the loss of reward or pleasure that is a major debilitating symptom in depression or melancholy. And it's a really amazing story that some of the very recent tangible headway in understanding the underpinnings, the causal mechanistic underpinnings of this syndrome and others like it, and the basic maladaptive and adaptive state transitions that happen have arisen from microbial organisms and the amazing proteins that they make. Highlighting the value, again, echoing a theme you heard earlier from Peter of the importance of basic science. In both neuroscience and psychiatry, as you know, we can use electrical and magnetic interventions, but they can't resolve individual cells or connections that are near each other. The key principle with optogenetics is this gives us the cell type specific control while maintaining the high temporal resolution. And you heard about the wonderful work on discovery and elucidation of the microbial opsin gene products, starting with Dieter Osterheld and Walter Stekenius, who identified the bacteria rhodopsins in the early 1970s. The halorhodopsins identified by Mukahara and colleagues, and Peter Hagemann's pioneering work in identifying the channel type microbial opsin gene family, and many others have discovered new members of this family in recent years. Now, the unique leverage that these bring is to give us the control we talked about, but it's not so simple as just conferring light sensitivity to a cell. That has been done even using microbial opsin genes as far back as 1994 in this paper from Gerard Bolt, where they were able to confer light-activated currents into yeast cells. But to consider taking these microbial proteins and creating a versatile, generalizable neural control strategy in animals as complex as behaving mammals is a difficult thing to consider. And indeed, it took about 5 years from 2004 to 2009 or so to get all the microbial opsin gene-related techniques working in mammals. And in 2015, Nature Neuroscience asked me to show the very first step along that direction, which I took in July of 2004. And so here's my handwriting depicting this very first step. And there was a broader vision in my small lab at the time. I wanted to find genes, and you can see some of them listed here, not just channel rhodopsin, but also potassium channels, wild type and dominant negative potassium channels, with the goal of conferring regulatable control of excitability in targeted cells. And so this was I had a very poor, small lab at the time, not much going on, and so I had very simple assays. I kept simple tallies of the cells that were activated by light, and my readout in this case was activation of the membrane depolarization recruited transcription factor, CREB, which gets phosphorylated on serine 133 in neurons in response to membrane depolarization. And this red stain is the antibody recognizing this nuclear readout of the membrane depolarization of the neurons in response to light. These are two neurons expressing the channel rhodopsin gene, illuminated and non-illuminated. And there were more cells that were positive, just barely statistically significant in the case where there had been illumination. And so this was enough to decide that this was going to work. It was at least had a chance of working. It was safe. It was tolerated. The neurons, fragile and vulnerable neurons, could express this algal protein in their membranes. And traffic it to the processes. And this was the first step. Of course, there was a lot more to do. This was our small group at the time, myself and the two graduate students, Feng Zhang and Ed Boyden. And they worked very hard for the subsequent year. I transduced a new round of cells, gave them to Ed, who did some very nice recordings and showed you could get high temporal resolution of the neural responses. I gave the gene to Feng Zhang, who was able to design very elegant viral strategies, as well as fiber optic based interfaces in order to make this preparation work in behaving mammals. This, in fact, was one of Feng's drawings from 2006, and it's basically identical to how we and everybody else does this sort of work now. So he's quite a good artist as well. Of course, big question that we faced was, is this actually going to work even in a non-surface brain structure in a mammal? Of course, all the discussions about psychiatry and so on, as well as basic neuroscience, are more or less moot unless you have a versatile strategy for accessing different parts of the brain. And this was the moment in 2007 when we first realized that this was likely going to work. This is a mouse where Feng had implanted on the right side of the animal's secondary motor cortex a optical fiber. This was a transgenic mouse expressing channel rhodopsin in chiefly layer 5 pyramidal neurons. And when he turned on the light, the animal began rotating to the left instantaneously. And when he turned off the light, as you'll see shortly, the animal simply stopped and went about its business again. And so this was the very first moment when we thought this actually had a chance of working. That we didn't learn too much scientifically from that experiment, but that was definitely an interesting moment. Of course, things moved very rapidly after that, targeting a deep brain structure using that fiber optic interface in the lateral hypothalamus. Feng and Antoine Adamantidis and Alex Aravenis and Luis de la Sella together were able to play in different patterns of activity into hypocretin neurons deep in the lateral hypothalamus and show that some patterns of spiking, but not others, were able to drive the state transition of awakening. And much more recently, and I'm going to, this is the last sort of historical slide, I want to tell you about some very recent work from this year and even some unpublished work, which I'll go to right after this. But we've done a lot of work in my lab on trying to sort out the neural mechanisms of adaptive and maladaptive anxiety and fear behaviors. In fact, a lot of this work was led by Abhishek Adhikari, who's joining you all as an assistant professor shortly. And one thing we were able to do, the fiber optic interface, we like its invasiveness because that gives us an opportunity to do a special kind of targeting, to recruit cells defined by their connectivity. And we would never give up the fiber optic even if we could. That sort of experiment is illustrated here. You can inject a opsin into a master control region in this case, which we found was recruiting different features of the anxious state, the bed nucleus of the stria terminalis. And we could recruit different cells defined by their outgoing projections by positioning the fiber optic in different downstream structures. And anxiety is a complex behavioral state. It has multiple features to it. There are, as we all know, there are heart rate changes, there are respiratory rate changes. It feels bad, aversive, and it carries with it behavioral effects as well, like risk avoidance. And you can see that in this sort of test, the elevated plus maze. This is a mouse spending most of its time in the so-called closed arms with the elevated walls and very rarely going out into the open arms, which are very exposed. And so this is apprehension in the absence of immediate threat. What Kay Tai and her colleagues in the lab were able to find was an anxiolytic, an anti-anxiety structure buried within the amygdala. And when the blue letters come on here, that's when she starts playing optogenetic control over a projection, a cell population defined by its projections. And she starts activating this pathway here, and to our surprise, for the first time, the animal is willing to include the open arms of the maze in its exploration. Nothing else changes. It still also explores the closed arms, and its speed doesn't change, but it's simply, as it's reporting its apprehension to us by its behavior, that is instantaneously and reversibly altered. And in this way, we can understand the causal underpinnings of both adaptive and maladaptive states, and a great deal of work has been done on that. Now, the features of this fiber optic interface not only give us the ability to have animals under our control while they're carrying out complex behaviors, even social or aggression-related behaviors, and carry out this projection to find targeting. But as we also found recently, we can use that exact same interface with some additional downstream optics to collect back information even from the very same animal. And just this year has turned out to be incredibly useful in giving us activity-guided and closed-loop optogenetic control. And so this is the first step in that direction, explicitly designed to look from the animal's point of view exactly like an optogenetic control fiber, the 400 micron diameter fiber we use all the time. But we're going to collect back light through the fiber as well and some downstream optics to help us resolve very small signals. And we can put in, for example, into dopamine neurons in the ventral tegmental area using our recombinase-dependent viruses that Feng also designed. And we can introduce genetically encoded calcium indicators and collect population-defined signals coming out. For example, have a mouse carrying out sucrose consumption. If you just put in a yellow fluorescent protein into those dopamine neurons and the animal licks on a lickometer, as the red dashes indicate, you see no signal at all. And that's because it's just a yellow fluorescent protein. But if you put in a GCamp, you can see these beautiful transients habituating sadly over time, as all pleasurable things do. But you can see the time-locked behavior deep in the brain in a brightly lit room of this animal carrying out this behavior. And so we can collect back a signal as well. And so this has allowed what's happened just this year is deep in the brains of freely moving animals to have activity-guided and closed-loop optogenetics where we can match the timing and magnitude of events occurring during behavior. The first thing I'll tell you about is more targeted to the timing aspect. This is work led by Kelly Zalikuski in the lab. And what she defined was a risk-taking rat model. There was a paradigm that she worked hard to develop and built on a long history of pioneering work in the foraging literature. She wanted to see, are there trait variables that control whether animals will take risks or not? What's going on in their brains right before they take a potentially risky decision or not? What predicts that, and what causes it? And so she set up a task where an animal can initiate many trials with a nose poke and two levers pop out. One is the safe lever. She always, the rat is guaranteed to get a small but reasonable reward, water reward upon pressing a lever and collecting the reward. The other lever is risky. Some fraction of the time, there's an enormous reward, but that's a rarity. Most of the time, you get something almost negligible, much smaller than what the safe lever can provide, and so that's the risk. And at the onset of each trial, then, the animal has a choice, and we can do many of these trials. We can see what happened on the previous trial, how that affects what's coming up on the next trial. And she did a lot of work. I won't recapitulate it all here, but she found a very interesting signal in nucleus accumbens D2 type dopamine receptors. And the timing of this signal and when it appeared was very interesting. She only saw it depending on what had just happened in the past, just with one trial previously, whether the animal had made a safe choice or risked and won or risked and lost. And it appeared just at the time when the next decision was being made. So if the animal's coming off a safe choice, gotten its small but reasonable reward, there was no signal in these cells. This is with the fiber photometry method and the genetically encoded calcium indicator method. Also coming off a risky choice and a win, there was no signal at the next decision time on average. But coming off a risk and a loss, at the time of the next decision, there was a prominent peak in these of activity in these D2 cells in the accumbens, perhaps consistent with a stop or break signal indicating that this was something that was very recently bad and not something to be done again. Of course, that's a very interesting correlation, but without causation, you don't really know what it's doing. So what Kelly wanted to do was to match the exact timing of this signal during the task, and the task had been designed robustly so that she could do that. And indeed, she found that this was a causal signal. Matching that signal in those cells, in that brain structure, in these same trained animals, she could instantaneously and reversibly change rats from risk-seeking to risk-averse. And it mattered, the timing mattered. It was worked at the decision period, but not nearly as well at other times in the task. So that's matching the timing. Now I'll tell you about matching the magnitude. And here, you already heard about some of this from Peter, but the Volvox channel rhodopsins turned out to be very interesting in allowing us to match magnitude and achieve closed-loop optogenetics. And in 2008, following together with Peter and his students, we were able to show that this Volvox multicellular green algae gives rise to a channel rhodopsin with a red-shifted action spectrum, and we were able to improve its currents with a variety of modifications, and by 2011, getting red light-driven spiking. Now, we had initially thought this would be useful, and it has been used in a number of cases as a combinatorial optogenetic control to give you access to two separate populations. But by far, what's been most interesting and useful about C1v1 was something we hadn't anticipated at the beginning, and that's its ability to be recruited by infrared, or in fact, a two-photon illumination, which is incredibly valuable in giving single-cell resolution control. And this was shown by Rohit Prakash in the lab in 2012. This is an experiment in a living mouse accessing by a loose patch clamp recording a cell in layer 2-3 of an awake mouse, and scanning, just raster scanning two-photon illumination just above a cell, not getting spiking, and within the cell expressing C1v1, getting spiking, and then just below not getting spiking again. And that's in the Z, the most challenging dimension. It was even better in X and Y. And this single cell and two-photon control has now turned out to be very valuable in combination with GCamp imaging. So you can do all optical experiments, as we and many others have done, beginning with this paper in collaboration with David Tank. And the co-expression of C1v1 and, for example, the GCamp 6 series allows this sort of thing. This is from Jim Marshall in the lab in visual cortex, co-expressing these two proteins, hopping from cell to cell at will, pinging them, and observing their calcium responses. Cell number is indexed here, and this is time, and this is intensity of the calcium response. And so we can do all optical experiments, and you can do it at the single cell level, as I'll show you more of, or at the population level using fiber photometry. And what Tina Kim and Sam Yang were able to show, again, just this year, was matching magnitude of naturally occurring signals using this feedback from the very same animal through the very same fiber. So here's the fiber optic in place. You've got dopamine cells in the VTA transduced with both a Volvox-derived channel rhodopsin and GCamp6. And you can give the animal a little reward, and the light blue trace shows this characteristic transient of that dopamine population response, and then she tries to match it in that same animal with the optogenetic drive of the Volvox opsin. And at one light intensity, it's too big, but she can back off in that same animal until she exactly matches the magnitude of the naturally occurring signal. It may seem like a small thing, but we wanted to do this for years and years and years in optogenetics, and really just this year, the tools have come together and it's become possible. And of course, you can do this at the single cell level too. Here, there's been a lot of work that we and others have put in on enabling spatial light modulators to play in a hologram of light with single cell resolution. And so we're doing that here, driving these two cells selectively. These are co-expressing GCamp and C1v1. And you can see all the surrounding cells as well. We and others have gotten to the level of many dozens of singly defined neurons controlled at once in the brains of behaving mammals. And so this is what you might call modern optogenetics. You know, you can be acute or chronic. You can go on any time scale you like. You can give excitation or inhibition. You can tune the magnitude thereof. You can have local or many targets across the brain. You can have, for example, up to 7 fiber optics across the brain of a freely moving animal, as Tina showed here. And you can target cells or cell types or projection types. And crucially, now we can match or modify, if we like, to test the importance of the timing and amplitude of activity in those very same components during behavior. All of this is activity guided. There's one step further, which we've just gotten to as well, which is true closed loop optogenetics. So in the very same animal, trying to feed back the information in real time. And this is now possible by integrating the Volvox opsins and GCamps as well, going from open loop to closed loop, just like a car cruise control. And Noah Young and Tina Kim have established this now just recently. This is unpublished data, but again, in a freely moving mice co-expressing these tools. This is the population response over time before they turn on their PID-based proportional integrative delay model for closed loop control. And they do that right here, choosing to fix the activity level at this dotted line. And you can see when they turn on the closed loop control, they're able to do that. And the looping time is now in the small numbers of milliseconds, 5 to 10 milliseconds. So this gives us, this is even using a genetically encoded calcium indicator. We can probably go faster if we have voltage sensors. Now that's one step forward. Another step forward relates to what you've heard some about already from Peter. Getting to the crystal structure of channel rhodopsin allowed us to see in 2012 that there's this dimer of 2 7-transmembrane proteins. Each has its own pore. And the lining of the pore, although it was a closed state structure, and we're still looking for open state structures, but the pore is crystal clear because it's lined with polar and indeed largely negative electrostatic negative residues. And this suggested a model of a pore function that Andre Berendt and Sue Lee in the lab tested. And their thought was, could we re-engineer, reupholster the whole inner lining of the channel and turn it from a cation channel to an anion channel. And they made hundreds of different mutations in an effort to try to achieve this. And they were able to turn the predicted surface electrostatics of the pore from largely negative, perhaps concordant with its cation selectivity, to largely positive, as shown here. And this came out back to back with a beautiful paper from Peter with different mutations, but same basic result. And this was an inhibitory, indeed, channel rhodopsin. It conducted chloride, and you could shut off spikes with blue light instead of driving them. And with inhibition, it's interesting, as many of the practicing neuroscientists will know, although there are cases where you want very brief inhibition sometimes, usually when you're delivering inhibition, you tend to want it over a longer period of time. You still want it to be temporally precise and reversible, but there are cases where you don't necessarily want to be delivering light for the entire time, over which you want effective inhibition to be recruited. And as you heard, back in 2008 and then in 2011, again, together with Peter, we were able to show that you could make tools for neuroscience that take this very fast deactivation to this very slow deactivation, and you can create bistable optogenetic tools. We had shown that for excitation, but we could port those same mutations onto the chloride channel and create very effective bistable inhibition now. And these have been used and shown to work in behaving animals. A final interesting point on the structure and the structure-guided engineering. A year after Peter and I published our chloride channel rhodopsins in 2014, in the summer of 2015, John Sputich's group identified a naturally occurring chloride-conducting channel rhodopsin. And it was very interesting. The predicted surface electrostatics of that one, GTACR2, was almost identical to what Andre and Sue in the lab had created using structure-guided engineering. And so this suggests that we probably do have a pretty good understanding of the pore structure and selectivity, but of course, nature got there first. And these are useful in behavior. And this has been shown, and this is again worked together with Peter and a number of other people in experiments looking at cued fear conditioning and in conditioned place preference. And I won't go into those for the sake of time, but it was very reassuring to see that these tools were powerful enough to be useful in freely moving animals. Now, I'll just 2 more slides as I conclude. One exciting thing, as you can see, is starting to integrate optogenetic control with cellular resolution activity imaging, and then a very recent thread is integrating this sort of set of data streams together with local and global anatomy. And we now have cases where we can start to register activity patterns of individual cells that we observe during behavior with local and global wiring patterns of those very same cells in that same animal after removal, dissection of the brain and removal. And this is also compatible with detailed molecular phenotyping of the cells as well. So this is where a lot of the field is going is bringing all these data streams together, operative in the same exact individual. Final slide is I'll just note it's very often asked, what is there a clinical implication for this work? And I definitely get that question a lot. It might be because I'm a psychiatrist, but it may just be people naturally, it's a very natural question to ask with all the problems and challenges and lack of ideally effective and safe treatments in psychiatry. Is there a natural impact of this field? And I and my colleagues, Peter and Gero, we're all basic scientists. We're not trying to drive a clinical translation ourselves. But a very interesting thread of work has started to come out, what you might call optogenetically guided clinical trials. And I would say the clearest example of this came out last year. Antonella Bonci, who is a very prominent drug abuse researcher, formerly at UCSF and now at NIDA, published a paper in 2013 with a very striking result. They were able to take cocaine-addicted rats that were very highly drug-seeking and were able to powerfully reduce that drug-seeking behavior, that cocaine-seeking behavior, with an optogenic stimulation of a particular region of prefrontal cortex. And this was such a compelling causal result. Rats certainly like cocaine. People certainly like cocaine too, and there's serious problems that happen as a result. But it's very hard to treat, as many of you will know. And we have non-invasive brain stimulation methods like transcranial magnetic stimulation. And I use this in depression for some of my patients as well, but it's not very effective, certainly not at the population level. And this is because we just don't know what the causal circuitry is for depression. There's a lot of real estate in the human brain to cover. Very hard to design. It's hard enough to get a clinical trial to work with just giving each patient one pill per day and forget about trying to map out in a large population pulse patterns, stimulus locations, recruiting plasticity or not. It's very, very difficult. But armed with causality, if to the extent there is correspondence, and often there is beautifully between experimental animals and human beings, then you've got a lever. What they did exactly that, so they guided their explicitly, and Anto was on this second paper too, the clinical trial, explicitly guided by the animal work. They delivered TMS to dorsolateral prefrontal cortex of a population of cocaine-addicted human beings, had an experimental group and a control group, and they observed a powerful, very surprising reduction in actual cocaine consumption and cocaine seeking in the experimental group. Obviously, a lot more to be done, but this is an example of an optogenetically guided clinical trial, and it's a very interesting concept. This was diagrams from a paper that Emily Ferenzi, who you've heard about already, wrote. And she mapped out actually some of the causal circuitry that probably underlies this reduction in reward-seeking behavior, which of course brings us back to anhedonia and more than 200 years ago, these age-old questions. How is it possible that a human being can no longer seek the rewarding stimuli that were once so motivating and pleasurable? And finally now, with the concepts of optogenetics and the microbial opsin genes, we're finally making some headway. And I want to thank all these amazing students and postdocs who I've mentioned along the way, our many collaborators, including Peter and many others, and our funding agencies as well. And I'm very grateful to have the opportunity to share this work with you. Thank you.